Welcome back to this next video, and in this video we are going to talk about the blood transfusion. Now this video is actually a continuation of the last two lectures where I told you about the multiple alleles and we were taking the ABO blood type as an example of the multiple allele. So if you want to know about the basics of the multiple alleles in the ABO blood type, you should see those videos and I'll share the link in the description. But this particular video is actually focusing on the blood transfusions. That which kind of the blood transfusion they are allowed and which kind of the blood transfusion they are not allowed. Now the key to understand this particular table is that if you are giving an individual a foreign antigen, the immune system of that particular individual is going to make antibodies against that antigen and there will be a clumping reaction. This is the key to understand this particular video. In the last video I've told you that when you talk about the ABO blood type, you are actually having four different kind of the phenotypes. You can have the O type of the blood, you can have the A type of the blood, you can have the B type of the blood, or you can actually have the AB type of the blood. So there are four different phenotypes when you talk about the ABO blood type. When you talk about the, say for example, let us first discuss about the O type of the blood. So if you have the O type of the blood, your genotype will look like this. You will be having the small I and small I alleles. That is going to make your genotype. We will come to this table in a while. If you give an O individual an O type of the blood, that means you are giving him or her own blood type. That means there will be no clumping. So I'll be representing the no clumping by a minus sign and clumping by a positive sign. So if you give an O type of the individual and O type of the blood, there will be no clumping because the blood types, they are the same. So this kind of the transfusion, that is allowed. But if you give an O type of the individual and A type of the blood, that means that the antigens that are present on the red blood cell of the A individual, they are foreign to this O person. And when that particular antigen is foreign to this particular person, the immune system of this person is going to make anti-A antibodies because the antigen is A. Therefore, the antibodies that are made, we call them as the anti-A antibodies. So if you give an O individual an A type of the blood, you are actually giving him or her a foreign antigen. So the blood is going to clump by the help of these antibodies. So you cannot give an O type of the individual an A type of the blood because there will be clumping. Similarly, if you give an O type of the individual B blood type, so the B antigen present on the red blood cells of the B type of the blood, that is foreign to this O individual, and the immune system of this O individual is going to make anti-B antibodies because now the antigen is B. So the antibodies that are made, they are known as the anti-B antibodies. So if you give an O individual a B type of the blood, you are giving him or her a foreign antigen. And when you give them a foreign antigen, the immune system with the help of the antibodies is going to clump the blood. So you cannot give an O individual a B type of the blood. Similarly, if you give an O type of the individual an AB blood group, so the A antigen is foreign and the B antigen is foreign, the immune system is going to make both of these antibodies and a very severe kind of the clumping reaction can take place. So if you look at this table, the O type of the individual can only get a transfusion from the O type of the individual. The other kind of the transfusion, they are not allowed for the O type of the individual. Similarly, when you talk about the A type of the individual, if the blood type is A, the A blood type can be represented by two different genotypes. It can be in the homozygous form or it can be in the heterozygous form. Now, if you give an A individual an O type of the blood, you are not actually giving him anything foreign. You should see my last two videos to understand why the A individual is not getting anything foreign from the O individual. So if you give an A individual an O type of the blood, you are not giving them anything foreign. Therefore, the blood is not going to clump. If you give an A individual an A type of the blood, that is his or her blood type, there will be no clumping reaction. But if you give an A individual a B type of the blood, that means that the B antigen is foreign for the A individual, and the immune system of this individual is going to make anti-B antibodies, and the blood is going to clump. If you give an A individual an AB type of the blood, the A antigen is okay because that is already present on the red blood cell of this surface, but as the B antigen that is foreign, that means that the immune system of this individual is going to make anti-B antibodies, and the blood that is going to clump. I hope you are now getting the idea how this table works. If you talk about the B individual, again the genotype can be in the homozygous form, or the genotype can be in the heterozygous form. If you give a B individual, an O type of the blood, you are not giving anything foreign to this individual, so there will be no clumping. If you give a B individual an A type of the blood, the A antigen is foreign for the B individual. The immune system of this individual is going to make anti-A antibodies because the antigen is A, and the blood is going to clump. If you give the B individual a B type of the blood, everything is okay. If you give a B individual an AB type of the blood, the B is okay, but the A antigen is foreign to the B individual. Therefore, this particular individual is going to make anti-A antibodies, and the blood is going to clump. And the last phenotype, that is the AB. And the AB phenotype is represented by this particular genotype. And if you give an AB individual an O type of the blood, that means you are not giving anything foreign to this particular individual, so there will be no clumping of the blood. If you give an AB individual an A type of the blood, that means you are not giving him anything foreign because the A antigen is already present over there. That means there will be no clumping. Similarly, if you give an AB individual a B type of the blood, the B antigen is already present in the AB individual, so there will be no clumping. Similarly, if you give an AB individual an AB blood, that is his blood type, this means there will be no clumping. So when there is clumping, that means that that particular transfusion is not allowed, and when there is no clumping, those particular transfusion, they are allowed. Now looking at this particular table, you can actually see that the O blood type is a universal donor, because if you give the O type of the blood to O individual, there is no clumping. If you give that to the A individual, to the B individual, or the AB individual, there is no clumping. That means you can give the O blood type to any of the blood type. And if you look at this particular table over here, the AB blood type is actually a universal recipient. It can receive the blood from the O, the A, the B, and the AB blood type. So therefore we call them as the universal recipient. Another important antigen present on the red blood cell of the individual, that is the Rh antigen, and I have a detailed discussion on that in my last video, but in this particular video I want to focus on the genetics of the Rh factor. Now the Rh factor, the Rh factor follows the law of dominance. Now that means that one allele is dominant and the other allele that is recessive. So the Rh positive allele that is dominant over the Rh negative allele. Now that simply means that if the Rh positive allele is dominant, the Rh positive allele is going to express itself in the homozygous as well as in the heterozygous condition, and the Rh negative allele will express itself only in the homozygous recessive condition. So if both of the parents, for example, they are Rh positive, that simply means that all of the offspring that you will be expecting, they will be having an Rh positive blood group. Say, because if this is the gamete from the mother, this is the gamete from the father, if they combine with each other, all of the time you are actually getting two alleles for the Rh positive factor. So all of the offspring that will be positive. Now if one of the parent is heterozygous, as you can see it over here, and one is homozygous, again these two individuals, they are homozygous Rh positive, but these two offsprings, they are heterozygous, but as the positive is dominant over the negative one, so their blood group will be Rh positive, because the Rh positive allele is dominant over the negative one, so it will express itself in the homozygous as well as in the heterozygous condition. Now, if both of the parents, they are heterozygous, the one parent is heterozygous, the other parent is also heterozygous, you can actually see that this is homozygous Rh positive, these two are homozygous Rh negative, but there are 25% chances of an Rh negative offspring as well. Because if this plus combined with this one, you will be getting this particular genotype. If this Rh plus combined with Rh negative, you will be having plus and minus. If this minus combined with this plus, so this is plus minus, but if this minus combined with this one, you will be having a 25% chance of the offspring with an Rh negative blood group. So if both of your parents, they are positive and your blood group is negative, that simply means that both of your parents, they are heterozygous for the Rh positive phenotype. If you talk about the Rh negative one, if both of the parents, they are Rh negative, of course all of the offspring that will be produced, they will be having an Rh negative phenotype. So the Rh negative phenotype is only expressed in homozygous recessive genotype. Like you can see that in this particular case and in this particular case, when both of the alleles, they are Rh negative, that means that in that particular case, the phenotype will be the negative one. Now this Rh factor, sometimes it creates an important medical issue. This can create a medical condition. For example, there is an Rh negative woman and the Rh positive man, and they are expecting a child. Now this Rh negative woman, for example, conceive a fetus whose blood group is Rh positive. Now that means that the mother is Rh negative, the fetus is Rh positive. That means that this positive Rh antigen is foreign to this particular mother, to this particular woman, because the mother is Rh negative. That means she has never seen an Rh positive antigen. So if the Rh positive antigen is presented to the immune system of the Rh negative woman, the negative woman is going to produce the antibodies against it. But during the first pregnancy, there is no problem. The reason is that the antibodies that flow in only one direction, from the mother into the fetus. And if the mother has never seen the Rh positive fetus, that means that she will not be transferring any antibodies against the Rh positive factor of the fetus. But when the birth takes place, during the birth is very messy kind of the process. So during the birth, the cells from the Rh positive fetus that enter the woman bloodstream. Now there is a mixing of the blood, and for the first time, the Rh negative mother, she have her immune system has encountered the Rh positive factor. Now she will make antibodies against this because the Rh positive factor is foreign to her immune system. Now when the blood of the fetus and the mother that mixes, we say that the mother or the woman has become sensitized, and she now make antibodies to fight the Rh positive blood cells, because her immune system is now exposed to the Rh positive factor. Now she make antibodies against this. Now this is a problem in the subsequent pregnancies. For example, in the next pregnancy, if the fetus again is Rh positive, so now the antibodies from the mother can be transferred to the fetus, can be transferred through the placenta into the fetus. And now as the mother have got the antibodies against the Rh factor, so the maternal antibodies, they are going to attack the red blood cells. And this actually creates a medical condition because in the first pregnancy, the mother immune system was not exposed, so she was not making any kind of the antibodies against the Rh factor which was present on the red blood cell of the fetus. But now as the women have sensitized because of her first pregnancy, the birth of the first fetus, now the the antibodies from the mother is going to attack the red blood cell, the Rh factor of the red blood cell of the fetus, and that can actually create a medical condition. Now, in developed countries, this is not a problem, because when the mother is sure that her blood group is negative and she can actually conceive an Rh positive fetus, they are injected with an antibodies against the antibodies of the Rh positive factor. So when these antibodies, they interact with the antibodies which are produced against the Rh positive factor, they are neutralized, so the fetus is no more at risk. But in developing countries, this is still a problem, because a couple, they have one normal child, and all the subsequent pregnancies, they are you can say they are not normal. The fetus is actually attacked by the Rh positive antibodies of the mother.